Hello friends, now we will start with part 3 of the summary of the book Brain Energy, written by Dr. Christopher M. Palmer, who gives insights into the origin of mental disorders and the methods of treating them. Dr. Palmer's theory can usher in potent therapeutic interventions for pervasive psychiatric conditions for which standard pharmaceutical approaches have proven minimally effective. In part 3 of the video lecture series, we will be studying the contributing causes that cause mitochondrial impairment and thereby contribute to mental and metabolic disorders. In part 1 of the video lecture series, we had studied about the mitochondria, the most important organelles in the cell, their functions and how mitochondria impact metabolic and mental health. In part 2 of the series, we had studied about how brain energy imbalance contributes to mental disorders. We had studied how overactive functions, cells being hyperactive or cells being underactive, or certain cell functions being absent, or developmental abnormalities in the neurons, or problems with cell maintenance. All these can cause mental disorders. Now in the contributing causes, we will be studying various contributing causes like genetics and epigenetics, like food, fasting and gut microbiome, physical activity, hormones, neurotransmitters and other metabolic regulators. Also sleep, light and circadian rhythms. Now first contributing cause is genetics and epigenetics. Now genetics means our genes and epigenetics means the expression, the process of turning the genes on or off, that is the gene expression. Thousands of genes, they confer tiny amounts of risk. In the Human Genome Project, which was undertaken, all the genes in the human body, they were studied, and the researchers tried to find out that which genes confer risk for which diseases. Now it was found that the gene called the DISC1, it confers risk for schizophrenia, bipolar disorder, depression, autism. It has been found within the mitochondria, and it has been known to influence mitochondrial movement, fusion and contact with other parts of the cell, and this affects the neuron development and plasticity. Then one of the strongest risk genes for mood disorders, CAC and AIC, it plays a major role in oxidative stress and mitochondrial integrity function. So this gene CAC and AIC, it is a risk gene for mood disorders. Then there is a gene called APOE gene, which increases risk for Alzheimer's disease. Now what the researchers found was that it is not always the abnormal genes which are responsible for mental disorders. Genes may confer tiny amount of risk, but the researchers found no direct relation of cause and effect, and they were disappointed that genes were not conclusively established as causes for mental disorders. Rather, it was the epigenetic mechanisms. Epigenetics means the process of turning the genes on and off. Now, it is the mitochondria which are controlling these epigenetic mechanisms, and the process of transferring risk for mental disorders from parents to children occurs through these epigenetic mechanisms. For example, when mothers are exposed to high levels of stress, then cortisol gets released, and this cortisol can affect the development of the fetus in the uterus. Also, in the Holocaust survivors, it was seen that the fathers had been exposed to a lot of stress, and their children and grandchildren had higher risk for developing mental disorders, and the fathers, they transferred the risk for these mental disorders through their microRNA to their children. So since the fathers had a lot of stress. So through the microRNA, the risk for mental disorders got transferred to their children. So through epigenetic mechanisms, the risk for mental disorders is getting transferred from parents to children. So mental and metabolic disorders. Now about the genes. We have seen two genes, DISC1 conferring risk for certain psychiatric disorders like schizophrenia, bipolar, depression, autism, and the other gene CAC and AIC conferring risk for mood disorders, and the APOE gene conferring risk for Alzheimer's disease. Now this APOE gene, it is coding for a protein called as apolipoprotein E, and this apolipoprotein E is related to fat and cholesterol metabolism. Now this apolipoprotein E, it appears to have direct effects on mitochondria, and the gene APOE gene that is coding for this protein apolipoprotein E. This gene, it comes in three forms: APOE 2, 3 and 4. And in the study, it was found that about 25% of people carry one copy of this APOE4 gene, and those which carry one copy of this APOE4 gene, they are 3 to 4 times more likely to develop Alzheimer's disease. And about 2 to 3% people, they carry two copies of this APOE4 gene, and these people are 9 to 15 times more likely to develop Alzheimer's disease. Then we have seen this apolipoprotein E. It appears to have direct effects on mitochondria. Researchers looked at people who had different APOE types and measured proteins that impact mitochondrial biogenesis dynamics, that is fusion and fission, and oxidative stress. Those with this APOE4 allele had lower levels of these important mitochondrial proteins, and these levels correlated directly with Alzheimer's symptom. Then another study, it looked at astrocytes. Astrocytes are important support cells in the brain. So astrocytes, they are a type of glial cells. Glial cells they act as support cells for the neuron. They are non-neuronal cells in the brain which act as support cells for the neurons. And astrocytes are a type of glial cells. So another study looked at astrocytes, important support cells in the brain, and found that APOE4 gene, it impairs autophagy, that is the cell repairing itself by recycling old and defective parts, replacing old and defective parts. That is called autophagy. Mitochondrial function and mitophagy. Mitophagy is the mitochondria repairing itself by replacing old and defective parts. So this APOE4 gene, it impairs the function of autophagy, mitophagy and mitochondrial function, thereby adversely affecting the metabolic health and leading to Alzheimer's disease. Now the good news is that the researchers found that giving a drug that stimulates autophagy, it could reverse some of the abnormalities caused by this APOE4 gene. Now this APOE4 gene, it increases risk for cardiovascular disease, some other mental disorders and epilepsy, that is seizures. But it appears to decrease the risk for obesity and type 2 diabetes. So this is two different things. APOE4 increases risk for heart disease, some mental disorders and epilepsy, but it appears to reduce the risk for obesity and type 2 diabetes. Now this APOE gene, it codes for apolipoprotein, we have seen, and this apolipoprotein E, it is found equally in all cells of the human body, and this apolipoprotein is primarily found in the astrocytes, that is the glial cells, a type of glial cells in the brain, support cells for neuron, and in the microglia. Microglia are also immune cells in the brain, and they are a type of glial cells. And this APOE4 gene, it results in a very slow and gradual decline in the function of astrocytes and microglia, which are support cells for the neurons in the brain. Microglia are immune cells of the brain. So APOE4 results in very slow and gradual decline in the function of these astrocytes and microglia cells in the brain, and these cells are more clearly related to cognitive symptoms. Now these astrocytes and microglia, if they decline in their function occurs, then cognitive function will also decline. So it is adversely affecting these two in the brain, that is the astrocytes and microglia are being adversely affected by APOE4 gene, thereby leading to adverse effect on cognition. So these parts of brain are ones that will wear down and begin to fail over time, resulting in specific symptoms. Once these begin to fail, then other mental symptoms of Alzheimer's will also start. Why? Because different regions in our brain, they are interconnected. So if one region is impacted, others will be impacted as well. So this research directly implicates metabolism and mitochondria as a common pathway to Alzheimer's. So we can see that this apolipoprotein, which is made by this APOE gene, it appears to have direct effects on mitochondria. So hence metabolism and mitochondria, they are a common pathway to Alzheimer's disease. We have already seen that mitochondrial dysfunction, we saw in the part 3, that we saw it in the part 2, that is the brain energy, in which we covered the brain energy imbalance, that mitochondrial dysfunction is leading to accumulation of beta amyloid, that is problems in cell maintenance are occurring because of mitochondrial dysfunction. It is causing accumulation of beta amyloid, which is leading to Alzheimer's. So mitochondria are directly related to Alzheimer's. They are a common pathway for Alzheimer's disease. Now we come to epigenetics, that is turning of genes on or off. Gene expression. Most of our genes are basically the same. However, what is clearly different is the expression of these genes. Now skin cells, brain cells, liver cells all have the same DNA. The DNA is the same. But all these are different, they function differently. Why? Because of epigenetics mechanisms. Epigenetics is responsible for making different cells in the human body different from each other because of different gene expression. The different cells are expressing different genes. Now skin cells are expressing different genes, brain cells are expressing different genes, and liver cells are expressing different genes. There are different ways that the body controls gene expression. Now body is controlling the turning on and off of genes in different ways. Now what are these different ways? Now see, one way is to modify the DNA itself. Now the DNA itself is modified. Modifying the DNA itself, this is the DNA, it is itself modified by applying methyl groups. Now methyl groups, they are applied to specific parts of the specific sites on the DNA, and these methyl groups, they influence which genes are getting turned on or off. We know that genes, they are segments of DNA. Genes are segments of DNA. So these methyl groups are influencing which genes get turned on or off. So by applying these methyl groups to specific sites on the DNA, it will influence which genes get turned on or off. And these methyl groups, they can be added or removed. They can be added or removed as needed. But at least at some sites, they appear to become more stable over time. So at some sites, they may become stable also. So this is the first way in which the body controls gene expression, by applying methyl groups to specific sites on the DNA. Now another way in which the body influences gene expression is through histones. What are histones? Now histones are the proteins. They are the proteins that the DNA gets wrapped around. So histones are the proteins that the DNA gets wrapped around. And these are proteins that DNA gets wrapped around, and they influence which genes in the DNA get turned on or off. So this is through histones. Second way in which the body influences gene expression is through histones, which are proteins that the DNA gets wrapped around. In addition to methylation and histones, there are many other factors involved in epigenetics. Now, what are these other factors? Other factors include microRNA. These microRNA are non-coding RNA molecules. Now, RNA, they are macromolecules. They are actually nucleotide. They have nucleotide. They are nucleotide sequences. Ribonucleic acid. Now, nucleic acid is made of nucleotide sequences. Nucleotide is made of a nitrogenous base, a pentose sugar, and a phosphate group. So, there are two types of RNA. One is the messenger RNA and other is the messenger RNA that code for proteins. Messenger RNA are coding for proteins. And the microRNA are actually performing biological functions, and some RNA directly perform the biological function, while other RNA, they code for proteins. They do not directly perform biological function. They code for protein which then perform the biological function. So microRNA are non-coding RNA molecules. So factors like microRNA, hormones, neuropeptides, and others, they also are involved in epigenetic mechanisms. Now we have seen three ways in which gene expression takes place, in which the body controls the gene expression. One is by applying methyl groups to specific sites on the DNA. Another is by means of histones, which are proteins that the DNA gets wrapped around, and these proteins influence which genes get turned on and off. And the third way is through factors like microRNA, hormones, neuropeptides, and others. Now, smokers tend to have DNA methylation of the AHRR gene compared to non-smokers. So, smokers have DNA methylation, so there is an DNA methylation of the AHRR gene compared to non-smokers. But if they stop smoking, then the change in methylation, it is reversible. So if they stop smoking, then this methylation of AHRR gene, it is reversible and the damage can be reversed. So this was about epigenetic mechanisms. Now mitochondria are regulators of epigenetics. We have seen that mitochondria, they influence which genes get turned on and off. Now mitochondria, we know that they are involved in calcium regulation, and that serves as an important off switch for the cells. And mitochondrial fusion, when mitochondria fuse with each other, they also send signals which can regulate gene expression. So mitochondria, they are regulators of epigenetics. They influence gene expression through levels of reactive oxygen species. So we know that mitochondria are involved in production and cleaning up of the reactive oxygen species. We have seen this in part 1 of the lecture series. And mitochondria also influence gene expression through levels of glucose and amino acid, and through levels of ATP, that is ATP to ADP ratio. These are methods through which the mitochondria influence the gene expression. Mitochondria, they can also they perform the function of sensing the level of glucose in the cells, how much glucose is present and how much glucose is needed. So mitochondria appear to control essentially the expression of all genes of a cell. Now about the womb environment, that is when the fetus is inside the womb of the mother. In the womb, the fetus is bathed in number of metabolic signals like food, oxygen, vitamins, minerals, mother's hormones, neuropeptides. And use of all these things influence the health of the fetus, like food, oxygen, vitamins, minerals, use of alcohol, drugs, prescription medications, and other factors. So if the mother is using alcohol, drugs, then it may have adverse effect on the fetus. And prescription medications, if the mother is taking any medication, that can also affect the fetus. Now there is this hypothesis called the thrifty phenotype hypothesis, which says that the babies who are deprived of proper nutrition in utero, so when the baby is inside the womb, if it is deprived of proper nutrition, then they are likely to develop obesity, diabetes, and cardiovascular disease later in life. So these babies are more susceptible to developing these metabolic disorders and even later in life. And what this hypothesis ignored was that these babies are also more likely to develop mental disorders. So if the baby is deprived of proper nutrition when it is in the mother's womb, then it is more likely to develop mental disorders later in life and double the risk of schizophrenia and antisocial personality disorder. So the risk for schizophrenia becomes double. Risk for schizophrenia and antisocial personality disorder becomes double, as well as increased rate of depression, bipolar disorder, and addiction. Now next we come to early life. Now when the child is in the womb of the mother, then there is the passing of metabolic factor from mother to children in breast milk. Now when the child is born, then in the breast milk of the mother, metabolic factors get transferred from the mother to the child. Then one such molecule which gets transferred from the mother to the child in breast milk, it is an important molecule called as NAD, nicotinamide adenine dinucleotide, NAD molecule. Now this molecule, it is a critically important coenzyme. It is a critically important coenzyme that can be derived from vitamin B3 or niacin. So vitamin B3 or niacin is a source for this coenzyme known as NAD. Or the body can make this coenzyme using the amino acid tryptophan from protein. So tryptophan is an essential amino acid, which means that the body cannot make it by itself. We need to derive it from food, from an external source. And this tryptophan is converted into vitamin B3 or niacin. It is a source of vitamin B3 or niacin. Now, this coenzyme, that is NAD, it can be derived from vitamin B3 or niacin, or the body can make it using the amino acid tryptophan from protein. Now, what are coenzymes? We know about enzymes. Enzymes, they are biological catalysts. Enzymes have got two components. One is the apoenzyme. The protein component of the enzyme is called apoenzyme, and the non-protein component of the enzyme is called as coenzyme. And B complex vitamins, that includes 8 vitamins in the B complex group, that is vitamin B1, B2, B3, B4, B3, B5, B6. There are a total of 8 vitamins including vitamin B12. B4 is no vitamin. So 8 vitamins in the vitamin B complex group. So all these vitamins, they are an important source for coenzymes. Now we have just seen that this coenzyme NAD, nicotine adenine dinucleotide, it can be derived from the vitamin B3 or niacin. And this amino acid tryptophan, it is a source for vitamin B3. Now certain foods like corn and maize, they are deficient in tryptophan. So this NAD, an important coenzyme, it is essential to mitochondria. So it contributes to mitochondrial health. It is essential to mitochondria for energy production. And it also plays a role in maintenance of DNA and epigenetics, that is gene expression. So this NAD is important for gene expression, maintaining of DNA and energy production. It is important to stimulate mitochondria for energy production. And low levels of this enzyme, that is NAD, low levels of this coenzyme NAD, they are known to impair mitochondrial function, and it can cause epigenetic changes, and it is associated with aging and many diseases which may include mental or metabolic diseases. Now there was a study done in mice. For the mother mice who got extra NAD, they had improved blood glucose control, improved physical performance, less anxiety, improved memory, reduced signs of learned helplessness, which is a marker of depression, and greater neurogenesis into adulthood. So better development of nerve cells. Now we come to intergenerational transmission of trauma. So how epigenetic mechanisms, they are responsible for transferring risks for mental disorders from parents to children. Now a study was done in the Holocaust survivors 1980 study. It showed that people exposed to trauma and their children have different levels of sensitivity to glucocorticoids. So people who are exposed to trauma and their children, they have different levels of sensitivity to glucocorticoids. Now these glucocorticoids, they are steroid hormones and they reduce inflammation and they are known to suppress immune system. They also affect metabolism and they contribute to the circadian rhythm as well. That is what time we sleep, what time we wake up. Their levels are high, found to be high in the morning and reduced throughout the day. And they are also used to treat diseases caused by an overactive immune system like asthma, allergies, sepsis, autoimmune diseases, etc. So this is about glucocorticoids. They are steroid hormones and they are known to reduce inflammation and suppress the immune system. Now the study says that people exposed to trauma and their children have different levels of sensitivity to glucocorticoids. In particular, exposure to high levels of cortisol in utero, so when the child is in the womb, then exposure to high levels of the stress hormone cortisol, they appear to program children, resulting in higher risk of developing mental and metabolic disorders later in life. So if the mother is exposed to a lot of stress, it leads to production of cortisol, which exposes children to greater risk of developing mental and metabolic disorders in later life. Then with the genetic and epigenetic revolution came the discovery that many of these people have differences in methylation patterns of the glucocorticoid receptor and other DNA regions, promoter regions associated with stress response system. More recently, it has been discovered that even fathers might be passing on their traumatic experiences through epigenetic mechanism in sperm, such as microRNA molecules which are known to modify gene expression. So fathers who are exposed to high levels of stress, they may transfer the risk of developing mental disorders. They may transfer the stress to their children by means of epigenetic mechanisms like microRNA. So their children will become more susceptible to developing mental or metabolic disorders in later life. The studies have now shown that sperm in both mice and men have microRNA that gets transmitted to offspring. So microRNA are epigenetic mechanism which can act as a method of transferring the trauma from fathers to their children. Now levels of specific microRNA 449 and 34 have been shown to be directly affected by levels of stress, dating all the way to early childhood experience of fathers. So fathers who had adverse early childhood experiences, they will transfer their traumatic experiences or their stress to their children through the microRNA, and these children will then have risk for developing mental and metabolic disorders in later life. Now low levels of this microRNA results in higher stress. So if this microRNA is in low level, then it can cause higher levels of stress. So fathers who are stressed, then through their sperms the number of microRNA transferred will be lesser, and this will increase the risk for children developing stress or mental and metabolic disorders in later life. Then fetal exposure to mother stress results in higher rates of learning impairment, depression, and anxiety later in life. In first few years of life, separating a child from his or her mother can result in higher levels of cortisol throughout life. So if a child is separated from his or her mother, then it can result in high levels of cortisol throughout life for the child. And severe abuse can result in lower levels of cortisol. So these are some conflicting findings. So if a child is separated from the mother in first few years of life, it can result in higher levels of cortisol for the child throughout life, and severe abuse can result in lower levels of cortisol. Both these states, they take a metabolic toll and can be tied directly to mitochondria, given that mitochondria initiate the production of cortisol. So mitochondria are directly involved in the production of cortisol. So higher or lower levels of cortisol than required can take a metabolic toll and can directly affect the functioning of mitochondria. It can adversely affect mitochondria. Now, what genetics and epigenetics tell us about causes and treatment of mental disorders? Now, the researchers, they could not find specific genes related to mental disorders, though some genes confer tiny amounts of risk, like we saw in case of DISC1 gene conferring risk for psychiatric disorders, or the CAC-NIC gene conferring risk for mood disorders, or APOE4 conferring risk for Alzheimer's. But there was no direct correlation because there was inability to find specific genes relating to mental disorders. No conclusive findings because it was more the role of epigenetic mechanisms in mental disorders, that is turning of genes on and off was more responsible for causing mental disorders. We now know that there are usually not abnormal genes that cause mental illness. So it is not abnormal genes that cause mental illness. It is much more likely that the transmission of mental illness from parents to children takes place through epigenetic mechanisms or gene expression. And the hopeful aspect is that most of these epigenetic mechanisms, they are reversible. They are reversible, that is, the adverse effects can be reversed through proper interventions. The effects of in utero stress, microRNA levels, and levels of NAD. We saw that NAD is an important coenzyme. So the effects of in utero stress, microRNA levels, and levels of NAD, they are reversible, sometimes through lifestyle interventions alone. So these effects are reversible, sometimes through lifestyle interventions alone. So lifestyle interventions can include changes in diet, changes in exercise, changes in sleep, reducing or like eliminating the use of alcohol, drugs, etc., or reducing of certain medications, or adjusting the dose of certain medications. All these interventions can reverse some of the adverse effects. People are not usually born with bad genes that make it impossible for them to be healthy. So this is a hopeful aspect, that it is not that people are born with certain bad genes that they are destined to suffer throughout their life. It is not bad genes, rather it is gene expression, and this gene expression can be controlled, it can be regulated through certain lifestyle interventions, which can even reverse the symptoms of mental disorders, which can even help the patients achieve remission from mental disorders. So this is the hopeful aspect. Now microRNA. What are microRNA? They are a family of small stranded non-coding RNA molecules. So microRNA are a family of small single-stranded. So they have a single-stranded non-coding RNA molecules. So these RNA molecules do not code for proteins. They don't code for proteins. And these molecules, they help the cells regulate gene expression. So these molecules help the cells in regulating gene expression. Now we have just seen that RNA are macromolecular machines found within cells that perform biological protein synthesis. Now, RNA are of two types. One is the messenger RNA, that is the RNA that code for proteins. So, messenger RNA is a single-stranded molecule of RNA that corresponds to a genetic sequence of a gene, and it is read by a ribosome. So, it is read by a ribosome. Now, what are ribosomes? Now, I will correct myself. I said that RNA are macromolecular machines that perform biological protein synthesis. This is wrong. It is not RNA. It is the ribosomes. The ribosomes are macromolecular machines that perform biological protein synthesis. And what is biological protein synthesis? They do the protein synthesis through the translation of messenger RNA. Now, what are RNA? RNA are nucleotides. So they are a chain of nucleotides. They are nucleic acids. And RNA can be of two types. One is the coding RNA, like the messenger RNA, which codes for proteins. And another type of RNA is the non-coding RNA, which does not code for proteins. So RNA is a molecule that is essential for most biological functions, and RNA are either performing the functions themselves. These are the non-coding RNA, and microRNA are a type of non-coding RNA, and they are performing various biological functions. And the other type of RNA are the coding RNA, which code for proteins. These are the messenger RNA. So microRNA, they are a family of small single-stranded non-coding RNA molecules that help cells regulate the gene expression. Now next we come to the analogy of three cars. We have already seen this analogy in part 2 of the lecture series on the summary of the book Brain Energy, and we talked about three cars A, B and C, which were of the same make and model. So these cars, they had the same blueprints or genes, but they were very different from each other. Car A, we talked that car A is in California, and it is not driven much by its driver, and the weather is very pleasant, and the car A is mostly in its garage, and sometimes the driver drives it, and it is mostly only one or two times a week. And so car A is in excellent condition, and naturally it will have a longer lifespan. Now car B. We talked that it is in another place called New Hampshire, where terrain is very hilly and the winters are very harsh. And now the car B, it is driven very often, and it gets used very often. Now when it is driving on hilly terrain, then it needs to switch on its four-wheel drive. When there are blizzards often in this hilly terrain, and now the driver of car B needs to switch on the wipers. And also it is driven at night, so the driver switches on the lights of the car. So it is used a lot, and so it is subjected to a lot of stress. So naturally it will require maintenance, and it will need to adopt certain adaptive mechanisms. So in case of when we compare it to humans, the adaptive mechanisms are mostly anxiety or depression. So if a person, when we compare it to a person, so if car B is engaging in certain adaptive mechanisms, it is nothing surprising since it is subjected to adversity. So if a human being is subjected to environmental factors which are adversely affecting the health of the human being, when the human being is subjected to, suppose, abuse or overwork or stress, then it is natural that that person will have depression, anxiety. So this, we can compare this person to car B. Now we come to car C. Now this car C, it is not in a place like New Hampshire. Here the weather is pleasant, weather is not unpleasant, and also the terrain is also level, it is not a hilly terrain. And car C, now the conditions are just like car A. So car C is in favorable environment and it is not driven very often either. But car C, what it is doing is, the driver of car C is switching on the wipers even when it is not raining. It is switching on the wipers even in the sunny weather. So this is abnormal. And the driver is switching on the four-wheel drive even on the level road. The driver is switching on the lights, is switching off the lights, and the driver is not switching on the lights even during night time. So when the car is being driven during night, driver is not switching on the lights. So this is again abnormal. So car C can be compared, and the car C is traveling at a speed of only 25 km per hour even when the other cars are traveling at a speed of 60 km per hour. So this is abnormal. So car C can be compared to a person with a mental disorder. So this car C, it is natural that it may meet with an accident. It will require frequent maintenance. If it is not given proper maintenance, it will meet with an accident and it will die an early death. So car C represents the case of a mental, when we compare it to a person, it represents the case of a mental disorder. Because in case of a mental disorder, the cells are not doing what they are supposed to do. When they are supposed to perform a particular function, they are not performing that function, or they are overactive or they are underactive. When they are required to stop performing a function, they are hyperexcitable, they are performing that function. Similar to the car C switching on its wipers even in sunny weather. So this was the analogy. Now car A, B and C are of the same make and model, so they had the same blueprints, that is genes, but they were very different from each other. Now what were the reasons for difference? What were the reasons for they being different? Reasons were difference in health, maintenance and longevity of the cars. Now the reasons for these cars being different was the environment and a dysfunctional driver, applying brakes at the wrong time or failing to use adaptive strategies when needed. Now, why were the cars different? Now, A was in favorable environment, B was in unfavorable environment, C was in favorable environment, or may have been in unfavorable environment, but it was not doing the right thing at the right time. So this is an example of a dysfunctional driver applying brakes at the wrong time or failing to use adaptive strategies when needed. And also the environment was impacting also, because car B was subjected to lot of stress, lot of adverse impact from the environment, so it could have a shorter lifespan, but the adaptive strategies that it employed, they were natural. So if a person is in adverse environment, in a very stressful environment, and that person is experiencing depression, anxiety, then it is natural. But if a person is in favorable environment and expressing depression, anxiety without any cause, then that person may have a mental disorder. So the two factors that were influencing, that were responsible for difference in health, maintenance and longevity of the cars was, one was the environment, another was a dysfunctional driver applying brakes at the wrong time or failing to use adaptive strategies when needed. So this dysfunctional driver can be compared to the mitochondria in our cells. So the cell can be compared to a car, and the dysfunctional driver can be compared to the mitochondria, because it is the mitochondria which are regulating the cells, which are controlling the functioning of the cell. They are influencing other organelles. So if the mitochondria are dysfunctional, they are doing the wrong things, they are doing things at the wrong time, they are not performing their task, then they are failing to use adaptive strategies when needed, or they are applying brakes at the wrong time. They are not stopping the cells when they are hyperexcitable. So this can cause problems. In human terms, it means that the primary cause of mental illness are not usually in our genes. So the primary cause of mental illness are not usually in our genes, but instead they are in our environments or the drivers of our cells, that is mitochondria. So primary cause of mental illness is not in our genes, it is instead in our environments, which can include hormones, neuropeptides, neurotransmitters, use of alcohol, drugs or medications, or in the drivers of our cells, that is mitochondria. Now, what makes the mitochondria dysfunctional? Why do the mitochondria become dysfunctional? Now, even with people, even for people with this APOE4 gene allele, which impairs the mitochondrial function over time, there is hope for healing. So there is hope. Not everyone with this gene develops Alzheimer's. It was found that increasing autophagy can lessen the problem caused by this gene. So mental disorders are reversible, and in case of Alzheimer's, we saw this APOE4 gene was causing problems. It was impairing mitochondrial function. But if you make an intervention which increases autophagy, that is the cell mechanism of repairing itself, then the effects of this gene can be reversed also. Now, mental illnesses, even ones like bipolar disorder and schizophrenia, are likely not due to genetic defects that are permanent and fixed. So it is not a death sentence, or it is not a death penalty or life imprisonment. Instead, these problems, they are reversible. They can be reversed if we carry out the right metabolic interventions. Now we come to the next contributing factor which affects the mitochondrial health and our mental and metabolic health. These are the chemical imbalances, neurotransmitters and medications. Now neurotransmitters can include serotonin, dopamine, GABA, norepinephrine, glutamate. All these are neurotransmitters. Now we know serotonin is playing a role in depression, in symptoms of depression. Dopamine, it is said too much of dopamine is causing symptoms of psychosis. Now GABA is playing an inhibitory role. GABA secretion inhibits cells that are hyperexcitable, and it prevents the symptoms of psychosis. The norepinephrine is helping in focus. So reduced level of norepinephrine is found in ADHD. Now glutamate is associated with manic states, high levels of glutamate, abnormal levels of glutamate and dopamine associated with manic states in the bipolar disorder. So these are about neurotransmitters. Then medications. Medications may include both psychiatric medications and metabolic medications. Now the brain energy theory, it offers new ways to understand neurotransmitter imbalances and the effects of medications. Mitochondria and metabolism explain the underactivity and overactivity or hyperexcitability of specific brain cells that result in imbalances of neurotransmitters and the problem of either too much or too little neurotransmitter activity. However, neurotransmitters also go on to produce their own effects in target cells. So neurotransmitters, they also go on to produce their own effects in target cells, resulting in stimulation of mitochondria or inhibition. So what happens is mitochondria are influencing the production of neurotransmitters, and neurotransmitters are also influencing the mitochondrial function, because mitochondria have got receptors for some neurotransmitters. We studied in the part 1 of the lecture series that mitochondria, they were involved in the production of neurotransmitters. Then the neurotransmitters were being stored in vesicles, and they would travel down the axon to the release site for neurotransmitters. Then they would be released at the target tissue, and then there would be reuptake of neurotransmitters. So mitochondria are influencing neurotransmitter production and regulation, and neurotransmitters are also affecting the functioning of mitochondria. For example, serotonin neurotransmitter, It is found to increase the ATP production from mitochondria. So there is a feedback mechanism between mitochondria and neurotransmitters. Now, mitochondria and neurotransmitters are in a feedback cycle. Mitochondria play a key role in the production of many neurotransmitters, including acetylcholine, glutamate, norepinephrine, dopamine, GABA, and serotonin. And we have already seen that mitochondria have receptors for some neurotransmitters directly on their membranes. So they have receptors for some neurotransmitters on their membrane. Example, they have benzodiazepine receptors and they have GABA receptors. And mitochondria, they have an enzyme. They have an enzyme called monoamine oxidase. And this enzyme is involved in the degradation and regulation of neurotransmitters like dopamine, epinephrine, and norepinephrine. So this enzyme is involved in breaking down or degradation of these neurotransmitters. So the antidepressants, what do? They are said to increase levels of certain hormones like dopamine or serotonin. So they inhibit the activity of this monoamine oxidase. So by inhibiting its activity, the neurotransmitters are not getting degraded, and so their levels are increasing. So this was about the monoamine oxidase that is involved in degradation of some neurotransmitters. Now we come to an important neurotransmitter known as serotonin. This is known for its role in depression and anxiety, and it has a very prominent role in metabolism and mitochondrial function. It is known to control appetite, digestive tract functions, metabolism, and nutrients. And about 90% of serotonin in the human body, it is actually located in the digestive tract and not in the brain. So we can see that gut is directly connected to serotonin. So 90% of serotonin in human body is actually located in the digestive tract. And recent research has shown a direct role of serotonin in regulating production and function of mitochondria within the cortical neurons. So within the neurons in the cortex of the brain, serotonin is regulating. Cortex is the outer layer of the brain, and serotonin is regulating the production and functioning of mitochondria in the cortical neurons. It is increasing their production of ATP. It is reducing oxidative stress. And serotonin increases mitochondrial function immediately, and it results in mitochondrial biogenesis. It increases the number of mitochondria. Serotonin is converted into melatonin. It is converted into melatonin, an important hormone in the regulation of sleep. So serotonin is converted into this melatonin, which is important for the regulation of sleep. And this melatonin plays a powerful role in metabolism. So this melatonin is playing a powerful role in metabolism. Serotonin is also a product of an important metabolic pathway called the kynurenine pathway. Kynurenine pathway. Serotonin is a product of this pathway, and this kynurenine pathway, it involves the fate of the amino acid tryptophan. So tryptophan amino acid, it goes through this kynurenine pathway, and when people eat protein that contains this tryptophan, it has many possible fates. Two important ones are getting converted into serotonin. It can get converted into serotonin, or it can get converted into kynurenine, which is a metabolite of the amino acid tryptophan. And this kynurenine, it leads to higher levels of NAD. We have studied about NAD. It is an important coenzyme for metabolic health. So this tryptophan is finally getting converted into NAD. And this NAD enhances our metabolic health. We know that this NAD is a coenzyme, and it is obtained from B complex vitamins. Now this tryptophan, it gets converted into niacin. It is a source for niacin, which is vitamin B3. Now NAD is a coenzyme which is critically important for metabolic health. Now NAD has a profound influence on health and function of mitochondria because it is essential for energy production and managing of electrons. Problems with this kynurenine metabolic pathway have been found in many psychiatric disorders. So this is important. Problems with this kynurenine metabolic pathway, which involves the fate of the amino acid tryptophan, have been found in many psychiatric and neurological disorders, including depression, schizophrenia, anxiety disorders, Tourette's syndrome, dementia, and others. Now we come to the next important neurotransmitter, GABA. GABA is known for its role in anxiety disorders because medications that increase GABA activity like Valium, Klonopin, Xanax, they produce a calming anti-anxiety effect. So GABA, it is produced by the cortical interneurons. And this GABA, what is it doing? It is inhibiting the function of hyperexcitable cells. And we know that hyperexcitable cells are causing anxiety. So GABA is inhibiting their function. So GABA has an anti-anxiety effect. Also, psychosis involves hyperexcitability. So GABA has an antipsychotic effect. However, abnormalities in GABA neurotransmission. So if the GABA neurotransmission is not proper, then abnormalities in GABA neurotransmission, they have been found in other disorders, including schizophrenia and autism. So in autism and schizophrenia, abnormalities have been found in GABA neurotransmission, leading to hyperexcitable cells. Now, mitochondria directly influence and sometimes control GABA activity. So mitochondria are directly influencing and controlling the activity of GABA. One group of researchers found that mitochondrial reactive oxygen species levels, they regulate the strength of GABA activity. So if reactive oxygen species, they are produced and also cleaned up by mitochondria, and the levels of these are also regulating the strength of GABA activity. And mitochondria actually store or sequester the GABA within themselves. So mitochondria actually sequester GABA inside themselves, thereby directly controlling the release of GABA. Now, research group demonstrated a more direct link between GABA, mitochondria, and mental symptoms. A study was done in flies and involved a rare genetic defect associated with both autism and schizophrenia. Researchers, they showed that the mitochondria actually sequester GABA within themselves. Thereby, they directly control its release. And when the process of sequestering of GABA by mitochondria, when this process was prevented by a genetic defect, then social deficits resulted. And when researchers corrected the GABA levels or functioning of mitochondria, the social deficits were corrected. So in this way, mitochondria are they are sequestering GABA within themselves. They are directly controlling the release of GABA. And when the process of sequestering of GABA by mitochondria, when it was prevented by a genetic defect in flies, then there was anxiety, there were social deficits. And when this defect was corrected, then again there was an anti-anxiety effect and social deficits were corrected. GABA doesn't just affect mental functions, it also plays a role in metabolic disorders like obesity. So GABA, apart from playing a role in mental disorders, is also playing a role in metabolic disorders like obesity. Now, one group of researchers, they found that GABA plays an important role in brown adipose tissue. Now, this is fat tissue, a special type of fat that gets turned on when you get cold, and also plays an important role in overall body metabolism. Problems with GABA signaling in this type of fat, that is the brown adipose tissue, they result in mitochondrial calcium overload, and they result in metabolic abnormalities found in obese people. So what they found is that GABA plays an important role in this tissue called as brown adipose tissue, a fat tissue, a special type of fat that gets turned on when you get cold and plays an important role in metabolism. And problems with GABA signaling in this type of fat, and they result in mitochondrial calcium overload, and they result in metabolic abnormalities that are found in obese people. So GABA is playing a role in obesity also. Then GABA is affecting the function of mitochondria. As we have seen that GABA is affecting the function of mitochondria, and mitochondria is also affecting the release of GABA. So there is a feedback cycle between GABA and mitochondria. So as we have seen that there is a feedback cycle everywhere. Now we come to another important neurotransmitter, that is dopamine. Now dopamine, it is released from neurons. Mitochondria are directly controlling its release. It is released from neuron, it binds to receptors, and it is usually taken back into the releasing neuron for another round. We have already studied in part 1 of the lecture series that mitochondria, they are directly involved in the production and release of neurotransmitters. Now the mitochondria, they are involved in production of neurotransmitters, then the neurotransmitters are stored in vesicles, then they travel down the axon of the neuron, and they travel to the release site, and then they are taken in by the receptors. They are taken by in by the receptors of the target cells, and then after their function is done, the reuptake of the neurotransmitters takes place into the axon. So this is the whole process. So dopamine is released from neuron, it binds to receptors, and it is usually taken back into the releasing neurons for another round. Some of the dopamine ends up inside the cells. Some of the dopamine, it also ends up inside the cells and needs to be managed by mitochondria. And so dopamine, most of it is taken back and taken back for another round. It is reuptake of the neurotransmitter occurs after its function is done, but some of it also ends up inside the cells, and it needs to be managed by the mitochondria. And mitochondria, they have got an enzyme called monoamine oxidase. We have already studied this, which degrades the dopamine. And to degrade the dopamine and to make this enzyme and break down the dopamine, ATP is required, energy is required, and this is provided by mitochondria. And for energy production, the mitochondria are stimulated to produce more ATP so that the enzyme monoamine oxidase can break down the dopamine or degrade the dopamine. A recent discovery showed that dopamine is directly involved in regulation of glucose and metabolism. So recent study has shown that dopamine is directly involved in the regulation of glucose and metabolism. Now there is this dopamine D2 receptor. Almost all the antipsychotic medications, they affect this D2 receptor. We know that this dopamine D2 receptor, they aren't located just in the brain, they are also located in the pancreas, and they play a critical role in the release of insulin and glucagon. So these receptors, they are also related in the pancreas. This is the liver, this is the pancreas. Pancreas is a leaf-shaped organ that is there next to the liver, beneath the liver. So these D2 receptors are also located on the pancreas, and these receptors, they play a critical role in release of hormones like insulin and glucagon. Insulin, we know it converts glucose into glycogen, and glucagon converts glycogen back into glucose. So these receptors play a role in the release of insulin and glucagon. It has long been known that antipsychotic medications, they can impact weight, diabetes, and metabolism. We know that antipsychotic medications, they have effects on the weight, diabetes, and metabolism of a person. And there is a possibility that insulin might be playing a direct role in the antipsychotic effect. Now, antipsychotic medications, they have effect on this D2 receptor, which is also present on the pancreas. Now, when they are affecting this D2 when the meds they are affecting this D2 receptor, then obviously they are affecting the release of insulin and glucagon, and effects on the release of this insulin, it can lead to metabolic changes. It can lead to metabolic changes, and these metabolic changes can result in an antipsychotic effect. So it may have nothing to do with the dopamine D2 receptors in the brain. The brain has also got receptors for dopamine D2 receptors, but the author is saying that the antipsychotic effect may not be produced by the D2 receptors in the brain. Instead, it is produced by the D2 receptors located on the pancreas, because these receptors are playing a role in the release of insulin and glucagon. Antipsychotic meds are affecting these receptors, so they are affecting the release of insulin and glucagon. So changes in the level of insulin and glucagon are causing the antipsychotic effect. So there is a possibility that effects on insulin might be playing a direct role in the antipsychotic effect. It may have nothing to do with dopamine D2 receptors in the brain. So metabolic changes are causing an effect on the mind. So metabolic changes like effect on the insulin and glucagon levels are causing an antipsychotic effect, which is an effect on the mind. So this is how metabolic changes are related to mental health. Now we come to insulin, which is a very important hormone. We know hormones are chemical messengers. They are chemical messengers that are produced by one type of cells, and they have an effect on other type of cells. So this is about hormones. Now we come to the first hormone, that is insulin. This hormone is critically important and it affects mental health. So first we come to, now people, diabetes is of two types. One is the type 1 diabetes, another is the type 2 diabetes. Now type 1 diabetes, the onset occurs usually in childhood, and this is an autoimmune disorder. This is because the immune system attacks the cells in the pancreas which are making insulin. So when the immune system attacks the cells in the pancreas which make insulin, so the cells get damaged and no longer make insulin. So insulin is not getting made by the pancreas. So in type 1 diabetes, insulin is not getting made in the pancreas because the insulin making cells have got destroyed. So this is type 1 diabetes, in which the onset occurs in childhood. And in type 2 diabetes is usually in adulthood. And this is due to insulin resistance. It means that either the insulin made by pancreas is not enough, either less insulin is being made, or when insulin is being made but the cells become insulin resistant, which means that insulin is unable to enter the cells. So cells become resistant to insulin, so the insulin that is made by pancreas does not benefit the cells. So it results in high levels of glucose in the blood, high levels of insulin in the blood, but the cells are not deriving any benefit from it. And the person may feel tired and hungry and may lack energy. So this is type 2 diabetes. Insulin resistant, meaning that insulin isn't working effectively to allow glucose to be used as energy source. So glucose is not being used as an energy source by the cells because the cells have become resistant to insulin. This is type 2 diabetes. It is adult onset mostly. Now emerging evidence over the past 15 years suggests that mitochondria are important regulators of insulin production and secretions. So this is very important. Mitochondria are important regulators for production and secretion of insulin. Now mitochondria, they are involved in glucose metabolism. That is how the glucose is broken down, how it is used as a source of energy. And mitochondria are also involved in sensing how much of glucose is available. So the mitochondria in the cell is able to sense how much glucose is available and how much glucose is needed. So in this way, the cell can take in the amount of glucose that is needed. Now, if the mitochondria is not functioning properly, then it is unable to sense how much glucose is needed. So it may not admit insulin into itself. It may become insulin resistant. So mitochondrial dysfunction may lead to insulin resistance. So mitochondria are involved in glucose metabolism and sensing how much glucose is available. They ramp up production and secretion of insulin as needed. So mitochondria, when they sense that glucose is less, so they may, and the mitochondria in the pancreatic cells, they may ramp up the production of insulin. They may initiate the production of insulin by the pancreatic cells. So in this way, mitochondria are affecting the production and secretion of insulin. Now insulin also affects the functioning of mitochondria. Insulin can increase the ATP production by mitochondria. Mitochondria have receptors for insulin on themselves. So in this way, it is a feedback loop between mitochondria and insulin. Both are affecting each other. There is a bidirectional relationship, just as there is a bidirectional relationship between the mental disorders and the metabolic disorders. Now, mitochondria are known to play a significant role in type 1 and type 2 diabetes. So, mitochondria play an important role in type 1 and type 2 diabetes, with some experts saying that mitochondrial dysfunction may be the primary cause for type 1 and type 2 diabetes. Now, a review paper suggests that mitochondria appear to be important to the cause, complication, management, prevention of both type 1 and type 2 diabetes. Now, insulin itself, it stimulates the mitochondria to produce more ATP or energy, and also stimulates mitochondrial biogenesis. So, insulin is going to increase the number of mitochondria. It stimulates the production of more mitochondria as measured in muscle tissue. Insulin earlier was even given as a treatment for mental disorders. So, earlier, the people who had mental disorders, they were given insulin as a treatment. Diabetes over time may develop even more mitochondrial dysfunction due to insulin resistance. So, in type 2 diabetes, we have seen there is insulin resistance. So, insulin resistance is causing mitochondrial dysfunction. Why? Because insulin stimulates mitochondria to produce more ATP. Now, insulin is not properly being produced or is not properly being used. So, this can lead to mitochondrial dysfunction. So, diabetics over time may develop more mitochondrial dysfunction due to insulin resistance. So mitochondrial dysfunction leading to insulin resistance, mitochondrial dysfunction leading to insulin resistance, and insulin resistance further aggravating the mitochondrial dysfunction. It is a vicious cycle. It suggests that insulin resistance can be both a cause and consequence of mitochondrial dysfunction. Now we come to insulin and brain health. Now, insulin is playing a powerful and direct role in the brain functioning. Insulin receptors are located throughout the brain. So, throughout the brain, we have these insulin receptors located on the cells in the brain. And they are involved in regulating whole body metabolism, appetite, that is feeding, reproductive functions, liver functions, fat storage, and body temperature. Brain insulin also modulates or regulates the neurotransmitter activities, that is activity of the neurotransmitters like serotonin, dopamine, glutamate, GABA, etc. And so brain insulin modulates the neurotransmitter activity and mitochondrial function within the brain cells. Changes in insulin functioning have been associated with impairment of neuronal and synapse formation. So if insulin is not functioning properly, then impairment of neurons may take place and impairment of synapse formation. So synapse, we know they are the gaps between two nerve cells, and they play an important role in the transmission of messages or nerve impulses. So these synapses may be adversely affected if there is problem in insulin functioning. So insulin is playing a role in the neurons. It is playing a favorable role, a beneficial role for neurons, and if insulin functioning is problematic, then neurons will be adversely affected. Synapse formation will be adversely affected. Now, insulin has been known to influence GABA, serotonin, and dopamine neurons specifically. So these neurons, for these neurotransmitters, they are specifically influenced by insulin. Now we know that GABA is involved in psychosis. Now low levels of GABA can cause psychosis, hyperexcitability, it can cause anxiety. Low levels of serotonin can cause depression symptoms. Too much dopamine is also known to play a role in psychosis. Now insulin resistance can occur in brain. Now when the cells of the brain become resistant to insulin, when they are not letting the insulin get inside them, then this can lead to neurotransmitter imbalances. Because insulin is playing a beneficial role for neurotransmitters. So when the cells become insulin resistant, then it can cause imbalance in levels of neurotransmitters, causing psychiatric disorders, causing mental disorders. It can lead to overactivity and underactivity of neurons. In addition to being located on neurons, insulin receptors are also present on the support cells. So they are located on the neurons, that is the nerve cells. They are also located on the non-neuronal cells. They are also located on the non-neuronal cells, which are the support cells, which support neurons. So these are support cells called astrocytes. Astrocytes are, we have seen, they are a type of glial cells, which are support cells, which support the neurons. They are also located on the astrocytes that play a role in providing energy for neurons. So astrocytes, they are star-shaped cells, type of glial cells that hold nerve cells in place and help the nerve cells function properly. So astrocytes are support cells for the nerve cells. And these cells, the astrocytes, they can affect mood and behavior. Now we talk about certain studies that have been conducted. Now the first study in rats. Insulin receptors were genetically removed. When the insulin receptors were genetically removed, it resulted in anxiety and depressive behaviors in rats. So insulin resistance would also have similar effects. Now second study shows that insulin resistance resulted in mitochondrial dysfunction, because insulin is stimulating the mitochondria to produce energy. So if insulin resistance occurs in the cell, then it will lead to mitochondrial dysfunction, and this in turn caused psychosis. Now the third study says that children with high insulin resistance beginning at the age of 9. So children who had high insulin resistance beginning at the age of 9, they had 5 times risk of psychosis and 3 times risk of schizophrenia and bipolar disorder before they would turn 24 years. So high insulin resistance in childhood, it increases risk for mental disorders at a later age. Now we come to Alzheimer's disease. Alzheimer's disease is also known to involve insulin resistance in the brain. Alzheimer's is often called as type 3 diabetes. It is also known to involve insulin resistance in the brain. There is evidence that the brains of people with Alzheimer's are not getting enough energy from glucose. So there is evidence that the brains of people with Alzheimer's, they are not getting enough energy from glucose. Why? Due to insulin resistance, because the cells are not allowing insulin to get inside them. So brains of people with Alzheimer's are not getting enough energy from glucose due to insulin resistance, and this results in mitochondrial dysfunction. So when insulin is not getting into the cells, it will result in mitochondrial dysfunction. Now we come to insulin as a treatment. From 1927 to 1960, insulin coma therapy was widely used in the treatment of serious mental disorders. So insulin was given as a treatment for serious mental disorders in earlier times. Clinicians would inject patients with large doses of insulin until they went into a coma, and this process was repeated a few times per week. Most reports from that era suggest that it was a highly effective treatment, at least in some people. At one point, it was the most widely used treatment for psychosis and severe depression, because we know that insulin is playing a direct beneficial role for neurotransmitters and hormones, and for correcting mitochondrial dysfunction, for mitochondrial health. So at one point it was the most widely used treatment for psychosis and severe depression in the Western world, but it fell out of favor due to the advent of psychiatric medication. So when psychiatric medications were introduced, insulin as a treatment was abandoned. Now, Alzheimer's disease. For Alzheimer's disease, researchers have been using intranasal insulin, that is insulin that is injected through the nose, in clinical trials for a few years now. One research study used intranasal insulin in 62 patients in bipolar disorder to see if it could improve their cognitive function over 8 weeks. So they used this intranasal insulin in 62 patients in bipolar disorder to see if it could improve their cognitive function over an 8-week period, that is over a 2-month period. The people who got insulin, they showed improvement compared to those who got a placebo. So people who got insulin, they showed improvement in the symptoms compared to the control group who did not get anything. More important for treatment, however, is measuring your insulin and blood glucose levels. So we need to conduct certain tests in order to measure our insulin and blood glucose levels to identify problems like insulin resistance. So we need to measure the levels of insulin and blood glucose in our blood so that we can identify the problems like insulin resistance, hypoglycemia, that is low blood sugar, or other issues. So only when we conduct certain blood tests to identify the problem, only then we can move forward for correcting the problem. Only then we can adopt certain interventions for correcting that problem. So first we need to measure the levels of insulin and blood glucose to identify these problems, like whether we are suffering from insulin resistance or hypoglycemia or other issues. There are many tests and tools available for this, like fasting glucose and insulin level test, oral glucose tolerance test, continuous glucose monitoring devices. So these tests need to be conducted to identify the problems. And once the problems are identified, suppose there is a problem of insulin resistance, then we can adopt appropriate interventions to correct the problem or to reverse the problem. Now, this was about the role of insulin and how insulin plays a role in mitochondrial health, and mitochondria also influence the production and regulation of insulin. Now, in the next part of the video lecture series, we are going to discuss about the role of psychiatric medications on metabolism and mitochondria, then about the other factors which influence our metabolism, like which influence our mitochondria and metabolism, like the other hormones like cortisol, thyroid, estrogen, etc., and about the role of food, the food fasting and the gut microbiome in influencing our mental and metabolic health. So all these are the other contributing causes for mitochondrial and metabolic health. So with this, we come to the end of this part of our video lecture series. Thank you.